Hello, General Microbiology. It's Dr. Welch, and let's do this first half of our study of Chapter 1 and discuss what the study of microbiology is and take some time to look at the organisms that the study of microbiology observes. So, microbiology, right off the bat, it's like I said in class today, it's the study of the itty bitty things. It's the study of living things that are too small to be seen by the naked eye. Many of these organisms that you're going to discuss this semester in general microbiology are disease-causing organisms, but please remember that many species of bacteria and many species of fungi have lots of economic importance and help us in cheese making, wine fermentation, beer making, bread making, tons of stuff. So please go into the science knowing that it's not all about diseases. So we want to kind of think about how it was these small, itty bitty microscopic forms of life were first discovered. And we want to think about this from the perspective of the tools that we use to observe these organisms, primarily the microscope. And so the discovery of the organisms that we study in microbiology are directly tied to the observation of soil or water samples, um, under some of the, or just with, I should say, some of the earliest microscopes. And the very first microscope was developed by Antony van Leeuwenhoek. And von Leeuwenhoek's microscope, as I was describing today in class, is truly little more than a glorified handheld magnifying glass. But it was a instrument that you see here in the lower right hand side of the screen that held the specimen in place so that the lens of this very primitive microscope could be focused on the organism that Van Leeuwenhoek was observing. And in chapter 1, they do a very good job of describing this. So again, if you have not read this first half of chapter 1, please go back and do so, and then maybe come back and revisit this lecture once again. So the organisms that you're going to be studying in general microbiology are classified as microbes or microorganisms. And there are many kinds of microorganisms, and we want to take a brief moment and think about where these fit in the taxonomic system that was developed by Carolus Linnaeus. So the Linnaean system of taxonomy is one that many people learn, oh goodness, middle school, high school. It's reviewed again in our Biology 101 and 102 sequence. But for some of you, this may be the first time you've had this since your secondary education in the school system. So this system of organization goes from very broad, huge broad categories of the domain and classifies organisms based on both their morphological similarities as well as their physiological similarities and genetic similarities, all the way down to decreasing levels of organization to where you're getting more and more specific. You're getting fewer and fewer organisms in each level of classification as you move from the domain down to the species. So this example that you're given here for the human and the ostrich, I just snagged an image off of Google Images. It gives you an idea of the terminology that's used. So for us, we are members of the domain Eukarya. We're in the kingdom Animalia, phylum Chordata, class Mammalia, order Primate, family Hominidae, genus Homo, and species Sapiens. So it's just a system that we use to organize biodiversity. And the course that you're taking is going to stick to the classification of organisms there on that kingdom level. We're going to touch briefly on domains, but for the most part, we're going to be working there on the kingdom level. But in the accompanying lab class, you are going to be required to know for that class and from some occasions that Dr. Pinchuk has in the syllabus, you're going to have to know the genus and species names of bacteria that he focuses the study on for the important role that they play in many diseases or from an economic importance standpoint. So as we, pardon my phone ringing there, as we think about the organisms you're going to study in general biology or general microbiology, excuse me, we're going to be looking at bacteria, the archaea, fungi, protozoa, algae, and a few of the small multicellular animals. We're also going to take some time and talk about viruses and prions. So I have those two separated off in a box because neither one of them are living organisms. These things are basically a chunk of DNA inside of a protein coat for a virus. For a prion, it is simply a protein itself, and they are parasitic. They require a host cell of an organism to be able to reproduce, to copy their DNA, and to basically cause the viral infection or the prion-based infection. More on those in a few minutes. But up here with bacteria, archaea, the fungi, the protozoa, the algae, and the small unicellular animals, these are all different kingdoms. You have kingdom bacteria. You have, yeah, you actually have domain bacteria, you have domain archaea, and then we get into kingdom fungi, kingdom animalia, kingdom plantae for the rest of these organisms. Protozoa, I'll add, are members of kingdom protista. That's a kingdom that is probably going to disappear as those organisms are absorbed into kingdom animalia, plantae, or fungi. So what was all that rambling about? You are working with organisms on a high level of classification. We're not going to go into phylum, class, order, family for the majority of things that you're going to be studying. Just remember what kingdom they belong to, and then for those that you're required, know their genus and species names. So the organisms that are classified as microbes or microorganisms, the majority of what you're going to study are prokaryotes, a few are eukaryotes. So I want to include a picture here of a prokaryotic cell as well as a eukaryotic cell to refresh your memory that a prokaryotic cell lacks a defined nucleus and it lacks those complex membrane-bound organelles, whereas a eukaryotic cell has that well-defined nucleus that protects the genetic material, and it has these more complex membrane-bound organelles like ribosomes. And if this was a plant cell, or is what we're going to study, an algae cell, it would also have chloroplasts. But do not think that prokaryotes are any less complex in their internal physiology. These organisms have photosynthetic pathways for those that are indeed photosynthetic. You also, all living things, from bacteria all the way up to human beings, have to perform some means of cellular respiration to produce adenosine triphosphate, or ATP, because once your capacity to carry out cellular respiration ceases, that's a death. So even though these prokaryotes are much, much more simple in their physical structure, their physiology is just as complex as that of a eukaryotic cell. I'm going to leave that for now. I know there are some lectures coming up in the future that get into the cell structure more in detail, and I don't want to name off organelles that you will not be required to know once Dr. Pinchuk or the adjunct professor takes over the rest of this course. So moving on, with prokaryotes versus eukaryotes, to summarize some things that I just said, prokaryotic organisms are single-celled or unicellular. They have a circular piece of DNA that is not contained within a nucleus. There are no membrane-bound organelles. These are very small organisms. They reproduce by asexual means of reproduction, but please do know that there are ways that these organisms maintain their genetic variation. One is by mutation, and then there are mechanisms by which prokaryotic cells can either acquire or exchange chunks of the DNA with similar cells and cells of the same species. So do know that, the mode of reproduction they're saying here is asexual only. There are ways to maintain that genetic variation, or we wouldn't have situations like antibiotic resistance, okay? We would not have the development of these new traits in strains of bacteria where that was previously absent. So the prokaryotes that we're going to study are the bacteria and the archaea. The eukaryotes you're going to study are the protozoans, the algae, fungi, and a few of the parasitic animals. Those can be either unicellular or multicellular. They have non-circular DNA that is held within the nucleus. They have those complex membrane-bound organelles like chloroplasts that are present in photosynthetic eukaryotes and mitochondria that are present in all eukaryotic cells. They are larger than prokaryotes, and these have the potential for being sexual. That's their primary mode of reproduction for the vast majority of the eukaryotic species, but there are some asexual species as well. So looking at each one of these groups of organisms briefly, the bacteria and the archaea, these are prokaryotic organisms. Their cell walls are used to differentiate them. And this is where some of the physiology comes into your early studies here within general microbiology. In bacteria, their cell walls are comprised of a very neat molecule called peptidoglycan. And Dr. Pinchuk will give you lots of lectures on peptidoglycan. It is a structural carbohydrate that has a protein component to it as well. This is what you're going to learn in your lab class, helps to differentiate gram-positive bacteria from gram-negative bacteria. It's the ratio of peptidoglycan to the lipids in the cell wall that differentiates gram-positive from gram-negative. Your archaebacteria, the walls do not contain peptidoglycan. As we've already said, these organisms reproduce primarily by asexual means of reproduction, but again, they can acquire new chunks of DNA from the same species by some mechanisms where they can exchange it directly with a living cell, or they can pick up released pieces of the DNA once a bacterial cell dies and it bursts. These can live singly or in pairs, chains, or clusters. Your, again, lab is going to give you a wonderful visualization of that. The bacteria typically grow right around body temperature, 25 degrees Celsius, which is below body temperature, up to 37 degrees C, which is body temperature. A few can survive a little higher than that. The archaebacteria, the archaeans, are usually found in very extreme environments. You have, oh goodness, thermophiles that like very hot environments, halophiles that like very salty environments. We could go on. But they are not known to cause disease. They're some of the most ancient organisms on planet Earth. So bacteria can sometimes be pathogenic, and those are the ones you're going to be most interested in. But please know that much of the role of the bacteria in the environment is as a decomposer, to break down dead organisms and return the nutrients that they contain back to the soil. And that's what many of the plants will use, or that is what the plants use, in addition to carbon dioxide from the atmosphere to grow. So, you know, I'm an ecologist, so their role as decomposers is very important in the cycling of nutrients that happens in ecosystems. So again, with the bacteria, these, for the most part, can be very helpful. They can provide nitrogen to plants. Some of the species are known as nitrogen fixers. Many of them that are aerobic in their means of, or not, excuse me, many of the ones that are photosynthetic is what I should say, will release oxygen to the atmosphere. There are many of them that are symbionts with you and help animals to digest cellulose. Actually, that is within ungulates. We cannot digest cellulose, but you have gut bacteria. This is why the probiotic yogurts and probiotic supplements are so popular with people right now, as they improve the digestive process. If you're having issues with that, or if you've had oh goodness, everybody's had a tummy bug, and then your guts are just torn up after you've had a stomach virus and your whole system's been cleaned out from one end to the next. That's where, once your system can tolerate yogurt, it will help to reestablish those gut bacteria more quickly. Some bacteria are used to make cheese, some can be used to make vaccines and antibiotics, and many, many are used to help degrade waste. Anyone that's got a septic tank, when you pour Rid-X down the commode and flush it down into the septic tank, you're adding bacteria to that septic tank to increase the rate of decomposition in the septic tank. But others can be harmful and can cause disease such as tuberculosis. We'll talk about staph infections. You'll cover anthrax, urinary tract infections. We could go on and on. The fungi, these are eukaryotic. They have cell walls. They include the molds and the yeast. Molds are multicellular. They can reproduce either asexually, or some of them will exchange spore or not spores. Some of them will exchange nuclei and reproduce sexually. Yeast are unicellular, and nearly all of their reproduction is asexual by budding. And so you're seeing yeast in the lower right hand corner of the screen, and you see those little bubbles kind of forming at the end of the larger cells, and that's where the yeast are budding and getting ready to divide in half. So an example of yeast that is absolutely important in terms of beer, wine, and bread making is Saccharomyces cerevisiae. That's the one that's used in beer fermentation as well as wine making. Candida albicans is a good example of a fungal infection, and Candida albicans is basically what causes yeast infections. Protozoans, again, eukaryotic, single-celled, similar to animals in nutritional needs and cell structure for the ones you're going to be studying. There are other members of this kingdom. These are the ones that are members of kingdom Protista that can be plant-like, and you also have some that can be fungal-like. They usually can move, and they move either by cilia, flagella, or what are called pseudopods. And again, you'll look at some of these under the microscope in lab. Algae, they're eukaryotic. They can be either unicellular or multicellular. They are photosynthetic. They have cells very much like plant cells, but they're unicellular, or for the unicellular form. And remember that plant cells have a cell wall, whereas animal cells do not have a cell wall. So those protozoans do not have cell walls, but here with the algae they do. Both of these groups classified as eukaryotes. The microscopic parasitic animals, Dr. Pinchak will go over some of the parasitic worms that like tapeworms, oh goodness, lots of different things that I can name off. You can Google a list of parasitic worms, and just don't always click over to Google Images if you don't have a strong stomach. The viruses getting into our critters, they're not even critters, are chunks of DNA with a protein coat, we should call them. Remember, as I said earlier, that viruses and prions are not living organisms, but they are things that have great, huge impacts on living organisms, and in particular from a disease viewpoint. So viruses, these are neither prokaryotic nor eukaryotic. They are not alive. They cannot reproduce by themselves. They simply are a protein coat surrounding that genetic material. And that genetic material can either be DNA or RNA. Really wild stuff. Viruses are very neat to study. Some examples: the flu virus, the common cold virus, the AIDS virus. There are a whole multitude of these that you're going to talk about later on in the course. And then lastly here with the prions, this is kind of the newest one to the bunch. Prions were linked to the role that they play in mad cow disease or Creutzfeldt-Jakob's disease. There's others. Kuru is another example of this. And prions are misfolded proteins that once they are in the brain of animals, they can cause other correctly folded proteins to misfold and lose their function. And so what you end up with during a brain scan is you end up with literally these holes that are formed by the areas of the brain tissue that are infected with a prion, and it yields one of the prion diseases or prion conditions. Again, to learn more about this, do yourself a favor and Google mad cow disease, scrapies. As I said in lecture, they're looking at similar proteins in their link to a lot of neurological diseases in people. This is one of the fastest growing areas of medicine. Really interesting stuff. So with that, we have finished our review of the first half of Chapter 1, and thanks for listening.